Okay, thanks, Megan. This is Aimee Fullerton speaking with the Northwest Fisheries Science Center. So first of all, thanks to Megan and thanks to the North Pacific LCC for the funding to do this and the opportunity to present this work with you all. And thank you all for tuning in right after lunchtime. So I'm going to start talking and give the context and some of the first results, and then I'm going to pass the baton to Se-Yuen, who is going to describe some results from the modeling that she's done, and then I'll take it back to do the wrap-up. I also would like to acknowledge the contributions by Christian Torgerson and Josh Waller. Okay, so natural thermal regimes are really variable over space and time, and that's illustrated pretty well with this graphic here. This is a 1 kilometer floodplain in the Sauk River in western Washington, and the 4 little orange dots are locations where water temperature was measured, and that's what's pulled out over here in the panels, the graphs. So what you see is the seasonal pattern as well as the daily pattern of variation in this groundwater channel is quite a bit dampened relative to some of these others, especially the main channel. So there's a lot more variability both over seasons and over time, or over days in the main channel. So there's a lot of variability in time and even in this very small space. And salmon and steelhead are adapted to this natural variability. Unfortunately, we humans have come along and done a lot to change thermal regimes. You might imagine things like big dams and how that can alter the patterns of water temperature. Also things like riparian clearing or anything that we have done that might have disrupted the surface and groundwater connections. And then climate change is already having a big impact on water temperature, both in summer and in winter. And we're seeing some pretty significant increases, at least for a lot of rivers, over the past couple of decades. So salmon are affected by water temperature at each life stage. When they start their lives as eggs in the gravel in the winter, and when they come out as little alevins, water temperature influences physiology and development rates. Then as juveniles, when they are moving around in stream systems and rearing, water temperature can influence their behavior and their ability to find food and avoid predators. It also has an influence on when fish decide to smolt and head to sea. And then after they spend some time growing big in the ocean, they have to come back up rivers to find their spawning grounds. And water temperature, particularly warm water temperature, can be a real problem for certain species depending on when they're coming back. So one of the things I'd like to impress upon you in this talk is that we should care about spatial patterns and water temperature because it matters to the questions that we are trying to ask about fish. So at really broad spatial scales, we can think about ranges where species are now, where they might be in the future, if things that are now suitable become too hot, for instance. And then at really fine spatial scales, we can care more about how water temperature influences the connectivity, especially among habitats that fish need to move between to complete their life cycle. And that's just illustrated here in this thermal image where you can see some really cold patches. They're quite small. So elevated temperatures are stressful to salmon across life stages. For adults, it can result in decreased fitness or straying or delay and even death. For juveniles and smolts, it can alter the growth rates and ecological interactions, such as evading predators. And for both, they might be more susceptible to disease if water temperatures are too warm. And it can influence the timing that these animals shift from one life stage to the next, so changes in phenology. So fortunately, salmon can and do seek cooler water. This graphic is from Matt Kiefer and his colleagues at the University of Idaho, and it illustrates that adult steelhead do use cold tributaries during upstream migration. The graph is showing you water temperature in the main stem Columbia River here in gray over the summer period, and these colored lines are the temperatures in the bodies of steelhead that are moving up the river. And so in this section, you can see that the fish temperatures are the same as the river temperatures, and then they hit this threshold of about 20 degrees, and now we're seeing the fish temperatures deviating from the river temperature, which means that these fish are pulling off into these cooler water tributary mouths to hold and then resume their migration later when things cool down. So these are adults. We also know that juveniles thermoregulate. This is a photo by Joe Ebersol, and what you're seeing here is you're looking top down on this whole gaggle of little tiny juvenile Omicus that are hanging out in this really small cold water patch. relative to the warmer habitat that's right beside it. So we know the cold water habitat is really important for these fish, that they're keying in on it and that they're using it. And the charge that we all have is figuring out what to do about managing for that cold water habitat, how to protect it, how to think about the kinds of actions that we do and how those actions influence the cold water habitat for fish. So there's already been some steps done for us. There's a primer for identifying cold water refuges that the EPA produced in 2012. It was authored by Christian Torgerson, Joe Ebersol, and Drew Keenan. That's available, and in fact, at the end of this webinar, I have the link to that if you're interested in looking at it. But we also need to think about how these patterns might change in the future as things get warmer. And really, ultimately, our goal is to try and figure out what is sufficient for cold water salmonids and how do we protect that habitat that they might need. And this is really a tricky thing to think about. It's something that the water quality standards and the people that are managing those are really grappling with, because it's not very straightforward to think about exactly what does sufficient mean for cold water fish. And I'm going to tell you right now that we're not going to completely answer that. It's something we're going to need to grapple with for a long time, I think. Okay, so the objectives of our research, pardon me, were to first use a large remotely sensed data set that I'll describe in a minute to characterize thermal heterogeneity in rivers, and that's really just a fancy word for patchiness in rivers. And then we'll think about how those patterns might change in the future given climate change, and then finally end with an illustration of what this might mean for salmon vulnerability to the loss of cold patches, at least during summer. So the data that we have available to us are these airborne thermal infrared remotely sensed river surveys, and they're shown in the black squiggly lines there within the center panel within the green area. The green area is the lower portion of the NPLCC boundary, so Washington, Oregon, and Northern California on the coast. And these were surveyed all by the same people. They're all larger rivers, and we don't have a lot of data for the small headwater tributaries. They were all collected on summer afternoons when things were meant to be stressful. So here's an image down here just showing you that you can see the difference in temperature from a cooler tributary moving into a warmer tributary. I'm also pointing out the location of the Snoqualmie watershed in Washington and the Siletz in Oregon. These are two case study basins that we dug a little deeper in with some of our analyses. So we can use these data to address spatial pattern questions across a variety of spatial scales, and I just wanted to sort of put in context the work that we're going to be talking about. If you did have enough data in a lot of tributaries, you could get at some of these whole network species range kind of questions, and we don't, and there are other types of data that are really good at answering that question that our data are not well suited to. We are better focused on these three lower scales, so moving down to a segment or whole river scale, looking at the shape of the profile as you move from downstream to upstream and how the differences among rivers might influence species diversity from river to river. We're really going to focus on this talk more at this scale. So this is thinking about the spacing and the size of relatively cooler patches at sort of an intermediate scale that fish might encounter as they're migrating upstream. You could also, with these data, look at very fine scale refuges, and we're not going to get into that today. So here's an example river profile. So I've got water temperature on the y-axis, and the direction is from downstream to upstream here. And you can see there's a lot of variability there in that water temperature that might be important to fish. So one way to go about characterizing thermal heterogeneity would be to chop this up according to temperature thresholds. So you could use whatever thresholds you want, but we chose some basic thresholds that are pretty standard in some of the regulations. So anything greater than 20 degrees we consider too hot. Anything less than 15 we would consider cool. And then when you look at it this way, you can start to think about where are these cooler patches, how big are they, how far up the river are they, etc. And so if we collapse all of this down into just a linear view here at the bottom, you can think about a fish moving from the bottom of the river up and where they're going to encounter those patches kind of in a linear way. And then we can look at that across all the rivers. And actually, these aren't all the rivers. These are just some of the bigger rivers in our study area. So I just want to pull out a couple main points here. there's a lot of warm habitat. There's a lot of orange on the screen. But there's also a huge amount of thermal diversity among and within these rivers. And this diversity is good for fish. So there's a lot of small, cooler patches, these little blue spots here and there, distributed throughout. But a lot of the blue you'll see is more in the upper end of the rivers for many rivers. Generally, these cool patches are large enough and within swimming distance for salmon in a lot of rivers, but certainly not all. There's some rivers that are just too warm all the way up, at least where we have data to survey. So what if we didn't have the spatially continuous data? What would we find about these kinds of metrics? So on the top, I'm showing you the raw data like I've already talked about, and how you would get these really small patches, perhaps as you cross a threshold. And if we aggregated that up to a resolution of, say, 1 kilometer or 3 kilometers, you see that these patches can get quite a bit larger. So when we look at these metrics, so how many patches are there, how big are they, and how far apart are they, starting with the raw data and smoothing all the way up to a 5 kilometer and beyond, you see this really nonlinear relationship here. And at a resolution of 1 kilometer and below is where these nonlinear relationships are strongest. So we're potentially losing a lot of very fine-scale spatial patterns and heterogeneity if we're not using this kind of spatially continuous data. So just something to keep in mind. Another way you could go about this, or maybe this is complementary, is to think about refuges in terms of relative refuges. So instead of absolute thresholds, we might have a fish moving upriver, and it's all warm, but here's a spot that's relatively warmer and relatively cooler. This is just one other way to consider, and I'm not going to talk more about it here, but we do a little bit in the report. A third way to think about this is to identify areas that are localized cooling or warming. And here we're just blowing up a section from the Ho River illustrating that. So the red is warming in a downstream direction, blue is cooling in a downstream direction. And this is something that could be really useful for trying to think about the drivers of these cold spots. So, what's causing that to be cold? It might be good to try and line that up to understand where this starts in the beginning. So one of the things we were asked to do with this project was to compare what we could learn with what has been produced by another NPLCC-funded project, and that's the Norwest project. So probably most people on this call have heard about Norwest already, but in case you haven't, just briefly, this is a product that Dan Isaac and his colleagues in the Forest Service have put together, where they got a lot of really empirical data from a lot of agencies, he calls it crowdsourcing, and put them all into one database. And then they used this spatial stream statistical model to predict water temperature throughout the entire region at a 1 kilometer resolution. So the predictions that you see here are for mean August temperatures. They've also got some scenarios for future climate change. And the spatial stream model includes some covariance structures that make it really valid on stream networks. So, okay, let me just back up for a sec, and I'm going to give you the punchline right now, and then I'll give you some of the details to back it up. The correspondence between our remotely sensed data and the Norwest data is very, very good at broader spatial scales, and perhaps a little less so at the really fine spatial scales. So to illustrate those broader spatial patterns, I've got a couple examples here from the 3 different states, so Washington, Oregon, and California. And what you're looking at are those long profiles of water temperature. The observed remotely sensed data are in blue, and the Norwest predicted temperatures are in black. And you see that these patterns are really, really well represented by the Norwest model. There's perhaps a little bit more variability in some of the remotely sensed data. And then the other thing that you'll notice is that the remotely sensed data are higher temperatures than the Norwest, and that's expected because Norwest is modeling a mean August, whereas our data represent a maximum summer temperature in most cases. Some days probably weren't maximum, for example. Okay, so we wanted to dig in a little bit more and see how they compare to finer resolution. And I just want to spend a minute just illustrating where we really have data to compare. So in our 2 case study basins, for example, these thick black lines are where we have thermal infrared data, and then all the thin gray lines are where Norwest predictions are available. And then these little green dots are the observations that Norwest used to make those predictions. So you can see there's really not a lot of places, at least in the Snoqualmie, that we have both sets of data, more so in the Silettes. One other note in this and other maps is that the tan color here is where anadromous fish can access. In the Snoqualmie, there's an impassable barrier there. So the other thing, and I already mentioned this, is our data represent maximum temperatures, whereas Norwest was a mean average August temperature. So there's a bit of a discrepancy there. When you correlate those two, there's definitely some strong relationships, but still quite a bit of uncertainty. So just note that, and what I'm going to show you next is using this relationship to scale the Norwest mean temperatures up to maximum so we can make some apple-to-apple comparison. And here I'm showing you in these two case study basins, so the Snoqualmie on the top, Silettes on the bottom, remotely sensed on the left, and Norwest predictions on the right. The purple dots are where we expect to see cold patches less than 15 degrees, and then the blue dot would be the most downstream patch that a fish would encounter moving from the river mouth in an upstream direction. So the trends you see is there's some differences here. So we don't have a lot of places that overlap in the Snoqualmie, but there's quite a bit of small cool patches that were picked up in the upper middle fork by the remotely sensed data that were not picked up by Norwest. We can't say anything about the tributaries. We don't have data there. And there's some differences down here as well. So we're not saying that it's not a great screening tool. It really is a great screening tool. We're just simply saying that one might need to get out in the field and follow that up with some field assessments to see if there's really cool patches there. Okay, moving on to think about what the future might bring. So if you were a fish, you might need some help navigating warmer water, and it's our job to sort of think about how to predict what those future patterns are going to look like. So I'm going to talk about the first two of these three methods that we use to think about future water temperature patterns, and then I'm going to pass the baton to Se-Yuen, who's going to talk about this third model here. So in one method, we just did a very simple assumption that patterns will just shift up. They're just going to get warmer. And we know this is a simplification, but there's a little bit of evidence, at least in these 6 rivers, and some of these aren't in the NPLCC, where we have repeat surveys between cool years and warm years. So you'll see that these spatial patterns, you can see the similarities from year to year, where it's cold in one year, it's cold in another. So at least there's a little bit of evidence that this isn't completely unrealistic for some rivers. And if we look at the results, on the left here, we've got the remotely sensed thermal infrared predictions, current day, and you've got orange is where things are really warm. In the middle here, we're saying if we just shifted everything up by 2 degrees, what would happen? So there's quite a bit more orange in this middle graph. And then the one on the right is showing you the places that would change. So the gray areas didn't change categories. They may get warmer, but they're still already warm. The green areas are showing places that used to be cool, and we're losing those. And the orange is showing places that are becoming really hot. Okay, so there's a lot going on here. Just to summarize this in general, couple metrics, moving from no change all the way up to a 3-degree increase in temperature. Yes, we would get more temperature warming by design. And what we're looking at here is the change in patch size and spacing. So not a whole lot going on because mostly patches are just redistributing on the landscape, but there are some places, some patches, the warm patches, where the upper tail is going to get significantly higher. So what that means is much bigger patches for some rivers where we lose the intervening cool patches, and correspondingly an increase in the spacing. So what this looks like in our 2 rivers, the Snoqualmie and the Silettes, under the past conditions, the thermal infrared data were taken in 2006 in the Snoqualmie and 2001 in the Silettes. Things were already kind of in this intermediate zone. And these long profiles, just to tell you, represent this portion here, this black line. That's where our data are in these 2 basins. So if we move things up by 2 degrees, we're going to warm up the main stem migratory corridor quite a bit in both rivers. In the Silettes, there's still some cool water in the headwaters. And then this is what that would look like on the landscape. So here's the past, here's the future. things are getting a lot warmer for fish. And again, we can't say anything about the tributaries. We just don't have data there. Hopefully there's some cooler refuges that the fish could pull off into if they needed to. And then this is the barrier where the anadromous fish can't get above. Similar down in the Silettes. So there's some existing cooler places that potentially could disappear in the future if things just shifted up. But we know that that's probably not going to happen in a lot of places, and we try to take a step forward in acknowledging that by using some statistical modeling that has some spatially variable climate variables. So these maps show you the predictions that change in, for example, air temperature between now and the 2080s, assuming a pretty high emissions scenario, greenhouse gas emissions scenario, the RCP 8.5. And you can see that there's a pattern here, right? The air temperature is going to get warmer in places and not as warm in others. There's also a pattern, the change in precipitation expected, and in the probability of precipitation falling as snow. So we use these three climate variables in a random forest model. So we fit our empirical data to these climate variables to predict a trend. And we pulled out the residuals because we know that there's a lot of stuff going on at a fine scale that's not included in our model here, and that are less likely to be affected by climate. So things like groundwater, surface water connections, and local riparian patches, et cetera. Then we predict the future trend by substituting in future variables, values for those three variables. We sum that back together with the residuals that we held out to get a future heterogeneity. And then this is what this looks like, at least for this river. So in the past, things were relatively cool here in the headwaters. In the future, they're going to warm up more, and not a whole lot of change going on in the lower part of the river. So some quick summaries of that from the historical, the data, the empirical data to our projection using the random forest model. One thing to note is that we expect to see a lot fewer patches in this intermediate category. And what's kind of more interesting to me is the size of patches here. So first of all, note the y-axis change. This is quite a bit larger than the graph to the left. So we will get some larger warm patches, and some of those are going to be really long. The spacing's similar overall, and again, that's more or less a redistribution issue where we've got some really long warm patches being, really long cool patches being chopped up or aggregated when they get warmer, and vice versa. Okay, so this is what this looks like in just a couple rivers, just to give you an example. The top bar shows what things looked like in the empirical data, and then the bottom bar is the future projection. So in this river, we had a lot of cool habitat here that got shrunk down to a tiny little piece, and same goes for this river. There's a whole lot of cold patches that just disappeared in the future. Okay, I'm going to pass this over to Se-Yuen, and she's going to tell you about a process-based model that we use as our third method.
Okay. The DHSM RBM is a coupling of DHSM, which is a hydrolyzing model, and RBM, which is a water temperature model. The water temperature model also includes the impacts of riparian trees on water temperature. The DHSM RBM is based on a mechanical understanding of how stream flow and water temperature respond to given climate, such as air temperature, precipitation, wind speed, incoming long and short waves, and to the given topography and vegetation. Therefore, DHSM RBM has been used to assess the impact of changes in climate and land use by using future climate and different land use scenarios. DHSM RBM produced the data at a relatively fine-scale spatial and temporal resolution of about 150 meters at 3-hour time steps, which is good enough to estimate the projected changes on cold water patches. That's why we choose the DHSM RBM as a process-based model for this project. DHSM RBM also have some limitations. One of them is it takes lots of time and requires lots of inputs to develop the model. Second limitation is it doesn't explicitly simulate groundwater. Before we look at the projected changes by the datasets from RBM, let's look at the calibration result first, because the well-calibrated result will give us more credit for its future projection. The top two panels show time series of stream flow and water temperature at the Salette River at the Salette. The bottom panel shows the distance water temperature profile from the downstream of the Salette to the north fork of the Salette River. As you can see, the historical simulations in blue reproduce the observations in black pretty well, except some locations. One of the locations is downstream of the Salette River in distance profile, where groundwater might cool down the water temperature, but this is not accounted for by DHSM RBM. Let's look at the calibration results for the Snoqualmie River. Again, the top two panels show time series of stream flow and water temperature at the Snoqualmie River near Snoqualmie. The bottom panel shows the profile from downstream of the Snoqualmie River to the middle forks of the Snoqualmie River. The TIL was surveyed around 3 p.m. for the upstream and around 6 p.m. in the downstream because there was a technical issue in aircraft which was used for TIL survey. So we used the same time period from the simulations. In other words, we used the 3 p.m. simulation for the upstream and 6 p.m. simulations for the downstream. Again, the historical simulations in blue reproduce the observations in black pretty well, except some locations. One of the locations is the confluence of north, middle, and south forks of the Snoqualmie River, where groundwater might also cool down the water. Up to now, we looked at that DHSM RBM was well calibrated for 2 case study basins. Let's look at the projected changes in water temperature. These two plots shows the spatial water temperature patterns for the Salette River for the historical period on left and for future period on right. To clearly see the changes, let's look at the difference between two. The projected changes in water temperature ranges from 0 to 4 degrees Celsius. The difference map also shows a spatial variability. For example, for the downstream of the Salette River, there will be substantial increase in water temperature. On the other hand, the upstream of the Salette River will have relatively small changes in water temperature. This result suggests that an approach using uniform increase across the basin might not be appropriate for some basins like the Salette River. Let's look at the projected changes in spatial water temperature pattern for the Snoqualmie River. The projected changes in water temperature will go up to about 10 degrees Celsius, which is a huge increase, especially for the fish. And this is about twice as large as for the projected changes for the Salette River. Let's look at the spatial pattern. For the warm tributaries such as Raging River, relatively little changes is projected, which is pretty good for the fish because lots of fishes move up to the raging rivers under current climate. On the other hand, for the historically cool tributaries such as North Fork, Middle Fork, and South Forks of the Snoqualmie and the Torch River, substantial increase in water temperature is projected. Let's think about that. Why the historically cool water patches will be more sensitive to the climate change compared to the warm tributaries, and why the Snoqualmie River will have a more increase in water temperature compared to the Salette River. The projected changes in stream flow provide answers for these questions. Let's look at the simulated stream flows. Blue shows the historical simulations for the 1990s. The red shows the future simulations for the 2080s. The solid line shows the on average of 30 years value. For the Snoqualmie River, no significant changes in stream flow is projected because historically precipitation fell as rain, so warmer winter temperature will not cause any form of precipitation. On the other hand, for the Snoqualmie River, substantial decrease in summer flows is projected because warmer winter temperature will cause precipitation fall as rain weather, resulting in substantial decrease in snowpack and summer snow melt. This result suggests that the substantial decrease in summer flow caused the more temperature increase for the location where precipitation used to fall as snow. Let's look at the projected changes in cold water patches. For the Sledge River, tolerable patches were predominant along the stream, and cold water patches were located at the upstream. By the 2080s, most of the tolerable patches will become warm patches, which is not good for the fish. But patches will still remain in the upstream. For the Snoqualmie River, historically tolerable patches will become warm patches. This is really bad news to fish. So based on these results, fish will have more challenges in the future as they move up to the upstreams, unless there are some cold patches along the tributaries. We are really interested in exploring how we mitigate these negative impacts, but we didn't have enough time for this project. One way that we could do this is we can adjust the riparian trees or land covers in DHS from RBM and then see how these factors affect the water temperature. I will pass to the Aimee so she can talk about how different approaches differently project in water temperature.
All right, thanks, Se-Yuen. So here we're putting side by side our three different approaches for looking at future water temperature patterns. The top is our simple shift method where we have no estimate of uncertainty. It just moves up some amount. The middle one is our statistical model, the random forest model, and then the bottom is what Se-Yuen just talked to you about. So one thing I would like to point out, because it's really painfully obvious right here, is that we don't have a whole lot of confidence in some rivers with the statistical model for projecting forward. And if you see the light blue lines around this dark blue line, this is the fit of the fitted model to those cove rates. So we have a pretty good fit. But when we're projecting outside of the bounds of what those relationships were based on, we don't have a lot of confidence. I mean, it's really saying things are going to get cooler in the main stem, primarily because air temperatures in the future don't warm as much along the coast. So probably we would put more of our confidence in this mechanistic approach. And so you already talked about the differences in magnitude here, where the sillets won't be quite as much warmer as what we're expecting to see in the Snoqualmie. Okay, moving on to the final objective, and this one's much shorter, I promise, just to try to think about what some of these changes might mean for fish. So I will give the example in the sillets because we have a lot of water temperature data there and we have a lot of fish there that we care about. There's a native summer steelhead population, there's a lot of ESA listed populations, and there are some TMDL plans in the works, in progress. So showed you this already earlier. This is cold water patches based on our assessment in 2001, just going back to our simple assessment of what things will look like in the future. But what does this mean in terms of specific species, specific life stages? For that, we need to consider when the fish are there and when we have data and where the fish are. So just as an illustration, this is summer steelhead who rear and migrate in this lower portion. This is probably mostly older juveniles and smolts maybe. And then here's the primary spawning and rearing up in the headwaters. So if we just look at the water temperature within the place where we know the fish are, for starters, and then also we can really only look at August. We don't have data for the rest of the year. We know that water temperature may stress these animals at other times of the year, but that's not something we can assess with the data that we have. So we can really just look at upstream migration and holding and a little bit of juvenile rearing. So vulnerability is a product of sensitivity and exposure and adaptability, which we didn't do, so I won't go into that at all. Sensitivity is just how a particular life stage in a particular species is expected to respond to water temperature, how stressed they may be. And this is primarily expert opinion based and, information out of the literature that we garnered. The meat of it here is in the E, that's the exposure equation. And the exposure equation says it's a product of these four variables. So P is the life stage present during August when we have data to look at it? If it's not, it's just going to default to 0. And then if it is, it's a product of how much that animal is using these habitats in August when we have data, how many cold patches are there, and how far apart are there. And then we can look at this for the different species and life stages under current and projected future conditions and see that and the scores range from 0 to 9. So some of these guys are getting zeros because they're not present during August. That doesn't mean that they're not going to be affected by warmer climates. They could be. So some of these scores are increasing. For a lot of species, this one doesn't change a whole lot. And I am not going to show you the results from the snoqualmie because things really don't change. They're already pretty warm for the fish, at least in the location where we have the data. So some takeaways. This is takeaways from this and from some other water temperature fish research that I've been involved in. Really, there's a lot of diversity out there in stream temperature. It's diverse across space and over time, which I didn't talk about at all today, but it's also significantly important. And these patterns are really important to the animals living there. So we've talked about a few advances in data and tools for thinking about how climate change may alter these thermal landscapes and affect fish. But really, the paper that I just got the proofs back today for a paper that summarizes a lot of this work, and we end the paper with 10 questions. So really, anytime you dig in and do some research, you come up with a whole bunch more questions that need to be future research priorities. So some management applications of some of the stuff that we've done are listed here, starting with water temperature regulations. There's work going on right now by a lot of people thinking about cold water patches and how do we manage for those. In the current regulations, there's criteria for sufficiently distributed cold water patches in places that are over 20 degrees, during fish migration season. And right now, the EPA and Oregon Department of Environmental Quality are working diligently on mapping the cold water refuges in the Columbia River and the Willamette River and trying their best to figure out what does this mean for fish. Are these sufficient, and where might be some places that they would need to do some more protection or conservation. And again, at the end of this talk, I have a link to their website if you're curious to learn more about that project. For salmon and steelhead, this is something that NOAA cares a lot about. And when we wrote our initial status reviews and recovery plans, we didn't have a lot of water temperature data available, so it was really hard to incorporate that into those plans. But given that there's more and more becoming available and we're realizing how important it is, hopefully this can contribute to that as well. Meanwhile, maybe it can suggest places where we would go out and prioritize some habitat restoration. It could be riparian, or it could be reconnecting to cold water off-channel habitats that the fish could use, etc. But again, this is a screening tool. People would need to go out in the field to really figure out what's going on. And finally, a lot of our plans, I think we're all starting to get on board with this, but a lot of our plans in the past didn't have climate considerations in them. So I wish I could leave you with some more specific things that we could do to manage thermal diversity in streams, but we'll start here. There's the two phases of planning and acting. For planning, we can do some characterizing of the kind of habitats that are out there now, sort of like we talked about, just as a baseline for thinking about where things need to be fixed. This second bullet, this is what we're all struggling with. What is sufficient for fish? Again, like I said, we didn't really answer that, and it's something I think is going to need to be dealt with on a case-by-case basis. But we can look at the water temperature data and information that we have and at least find places that look like they could be impaired during certain times and focus there. And then again, consider what will change in the future. What can we do now? We might want to prioritize actions according to what is the most threatening and risk for fish. For instance, migratory pathways during summer. If we don't get the adults up to the spawning grounds, I suppose nothing else matters. Implementing conservation activities that do no harm, like planting riparian restoration and allowing fish access to cold water habitats within the floodplains, if possible, anywhere, is certainly going to help things. And then circling back to see, how effective these actions were and how the fish are actually responding to them. Okay, so thank you very much to the North Pacific LCC for the funding and the opportunity to do this project, and to Emily Alfred, Joe Ebersole, Russ Fox, Dan Miller, and Ashley Steele for all their good, hard work and thoughts in the project as well. And while we open it up for questions, I just wanted to leave you with this additional resources. I believe Megan's going to post this PDF online so you can refer back to it later, but I mentioned the EPA Cold Water Refuges Project and the Northwest Power Planning Council. Leslie Bach gave a presentation on cold water habitat recently, and you can get to that presentation from this link. Here's the EPA primer for identifying cold water refuges that Christian Torgerson and others wrote. The first citation here was just accepted and should be out soon, and that summarizes a lot of the work we talked about today. There's a couple other papers here that you might be interested in, and including one that Dan Isaac just got out a couple days ago too, about the Norwest project. Okay, so we're ready for questions and feedback. Thanks for your time.
Great, thank you so much. So just a reminder for everyone on the phone, if you'd like to ask questions to our presenters, please use the chat function and you can send them to NPLCC staff, and then we'll ask the presenters for you. And as a reminder, all of the resources that Aimee just shared, as well as a recording of this webinar and the presentation slides, we will send that to everyone after we gather it all together and get it posted online. So we have some questions. The first one is from Eric Doyle. First, he says, thank you for sharing the interesting study methods and findings. And his question is, in my experience, vertical thermal variability, surface to bottom, is not well represented by FLIR data collection, particularly in summer. In certain watersheds, example the Okanagan, it's apparent that there are cool water patches near the channel bed that aren't reflected in the FLIR data. How well do the presenters think their methods would capture the aspect of variability, and how adaptable are their models to new information?
Okay, so I'll take that one since the remotely sensed data are the part that I worked with. Yes, they can be missing some of that vertical stratification. Christian Torgerson, who's really the expert on this kind of data, his experience, and Russ Fox, the guy who collects all the data, they tell me that for most of these rivers, they are well mixed and that they don't see that stratification being an issue for most places. I can't speak to that specifically myself. We certainly know there are places, and there are places in some of the bigger, warmer rivers, like the Eel River down in California. We know we're missing some of those deeper pools, absolutely. So that's definitely something to keep in mind. Again, it's a screening tool. In terms of being able to adapt to including new information, absolutely. I mean, any ideas that you have, please throw me an email. I'd like to think more about how we can make this better and more useful.
Thanks. So Eric added that the Snoqualmie is one watershed where vertical thermal variability may occur.
And perhaps that's why we see such a big homogeneous hot stretch. I hope you're right. We have actually, incidentally, Ashley Steele and I have about 40 sensors out there, and we've built a spatial stream network model similar to what Norwest does. So in this watershed, we're going to have the opportunity for really digging in and seeing how well these patterns hold up across all of our models. and I didn't get to talk about that at all today.
Thank you. So as of right now, oh, here comes the question. So this is from Kim Hyatt. Process models, although most useful, tend to be data hogs. So what portion of rivers have a high enough data density of the right types to apply these process models to?
How many rivers do we have enough data for running the DHS-BM RBM?
It depends on each watershed, but if each watershed has long time series of the water temperature data and some of the special pattern of the water temperature data, I think that we are able to implement the DHS-BM RBM. Especially for the DHS-BM, the hydrologic model, we have lots of good stream flow data from the USGS gauges, so it is a pretty easy part to implement the DHS-BM hydrologic part for any basin. But for the water temperature, sometimes we have a very difficult time to get those observed temperature data. So it totally depends on the temperature, whether we can get enough temperature data or not.
Thank you. So no questions currently, and just a reminder, if anyone still has some questions, you can use that chat function and send them to NPLCC staff.
And please reach out if you have thoughts or ideas. My email address is really easy to find. It's my first name dot last name, so aimee.fullerton@noaa.gov. I can put you in touch with Se-Yuen. Hers is a little more complicated.
Thanks, Aimee, and I'll make sure that I send both of your emails out with a follow-up with all the materials as well, in case anyone comes up with any questions in their sleep tonight.
Okay.
So no questions currently, but I'll keep the floor open for a few more minutes. We've got 9 left in total, so feel free to send some. And thanks again to our presenters and everyone that joined us today. We got a thank you from Lucas Nip.
Thanks for listening.
All right, seeing no more questions coming through, we'll go ahead and end it here. But if anybody thinks of any, just feel free to get in touch with either of our presenters or anyone from the NPLCC, and we can get you to the right people. And thanks again. Have a good afternoon.
Okay, bye.
Thanks, everyone. Bye.
Bye.
The leader has disconnected. This call will now end.